Today, we will discuss enzymes, the biological catalysts that make life possible. Let's explore how these incredible molecules speed up chemical reactions and play a vital role in everything from digestion to cellular respiration. What are enzymes? Enzymes are biological catalysts, specialized proteins that accelerate biochemical reactions within cells. What makes them remarkable is that they are not consumed during these reactions. This means they can be reused repeatedly, making them incredibly efficient. They work by lowering the activation energy required for a reaction to occur. Without enzymes, many of the chemical reactions essential for life would happen far too slowly to sustain it. In short, enzymes are the unsung heroes of cellular function. Remember when we studied proteins and learned that their shape determines their function? That principle is especially true for enzymes. Each enzyme has a unique shape that allows it to bind to a specific substrate, the molecule it acts upon. If the enzyme's shape changes, it may no longer bind to its substrate, rendering it inactive. Take phosphofructokinase, for example. This enzyme plays a key role in cellular respiration, catalyzing the conversion of fructose 6-phosphate into fructose 1,6-bisphosphate. It's just one of many enzymes working behind the scenes to keep our cells energized. Enzymes are crucial because they reduce the energy barrier, known as activation energy, that must be overcome for a reaction to proceed. This dramatically speeds up the rate of reactions. While increasing temperature or changing pH can also accelerate reactions, these changes can denature proteins, including enzymes. Denaturation alters the enzyme's shape, making it inactive. The graph here illustrates the difference between a catalyzed and a non-catalyzed reaction. Notice how the catalyzed reaction requires significantly less energy, highlighting the efficiency of enzymes. Naming enzymes. Here's a helpful tip. Most enzyme names end in ase. If you know the substrate, you can often guess the enzyme's name. For instance, lactase breaks down lactose into glucose and galactose. Similarly, amylase acts on amylose, a starch molecule. This naming convention makes it easier to identify enzyme functions. Let's talk about how enzymes work. The induced fit model explains how enzymes interact with substrates. Initially, the active site of the enzyme isn't a perfect fit for the substrate, but as the substrate approaches, it interacts with the enzyme's amino acid side chains, causing the enzyme to change shape slightly. This adjustment allows the enzyme to better accommodate the substrate and catalyze the reaction efficiently. You might have heard the lock and key analogy to describe enzyme-substrate interactions. While it helps explain specificity, it's not entirely accurate. Why? Because a lock doesn't change shape when a key is inserted, but enzymes do change shape to fit their substrates. That's why the induced fit model is a more accurate representation of how enzymes work. So, be ready to challenge the lock and key analogy when discussing enzyme mechanisms. Some enzymes need a little help to function properly. These helpers are called cofactors and coenzymes. Cofactors are inorganic, non-protein molecules such as zinc, iron, and copper. They can bind to the enzyme's active site or to the substrate, helping form the enzyme-substrate complex. For example, the enzyme lipoxygenase contains iron molecules which assist in metabolizing polyunsaturated fatty acids, essential for both plant and animal cells. Coenzymes, on the other hand, are organic non-protein molecules. They're essential for enzyme function and often derived from vitamins. Two key coenzymes are NAD+ and NADP+. These molecules play a critical role in cellular respiration and photosynthesis, respectively, by carrying electrons during these processes. We'll explore these coenzymes in more detail when we study energy transformations in cells. Enzymes can also be inhibited, meaning their activity is reduced or stopped. There are two main types of inhibition: competitive and non competitive. Competitive inhibitors resemble the substrate and compete for the active site. When they bind to the enzyme, they block the actual substrate from attaching, preventing the reaction from occurring. This type of inhibition is like someone sitting in your seat before you get there. They're not supposed to be there, but now you can't sit down. Non competitive inhibitors work differently. They bind to a site on the enzyme that's not the active site, called an allosteric site. This binding causes the enzyme to change shape, which in turn alters the active site. As a result, the substrate can no longer bind and the enzyme becomes inactive. Think of it like someone bending your chair so it no longer supports you. It's not about blocking the seat, but changing its structure entirely. Cells need to regulate chemical reactions to maintain balance and efficiency. One way they do this is through allosteric regulation. Some enzymes have allosteric sites, receptor sites located away from the active site. These can bind either activators, which keep the enzyme active, or inhibitors, which render the enzyme inactive. This regulation ensures that enzymes are only active when needed, helping cells conserve energy and resources. Enzymes aren't just important in biology, they're also used in industry and commerce. From making cheese, alcohol, and lactose-free milk to producing soap, paper, and clothing, enzymes help break down substrates into useful products. Their ability to catalyze reactions efficiently makes them invaluable in manufacturing and food production. There are lots of real-world examples of enzymes in action.